I would like to highlight neurofilaments as a biomarker for multisystemic scars and ARCAs and take SCA3 as an example. And I want to focus both on human as well as mice work. And the work I'm going to show is actually only partly my work. I would like to really acknowledge Carlo Wilke from my group and Janette Hübner-Schmidt from Tübingen, because they did actually most of the work. So what are neurofilaments and why is it promising? Neurofilaments are part of the cytoskeleton, in particular of the long axons. And whatever type of damage hits your axon gives you a release of the neurofilament. So even if you're a soccer player, had the ball, this will lead to a release of neurofilaments. So it's in no way specific. Whatever puts damage to your axon, it will lead to a release of your neurofilaments. When we are going to study two neurofilaments, neurofilament light chain and neurofilament heavy chain, they differ in their protein weight and by the fact that neurofilament heavy chain does have a phosphorylated component. So this is why we call it phosphorylated neurofilament heavy chain. And we are going to analyze this by the SCA. The SCA stands for ultra-sensitive single molecule array. And it works a little bit like a digital ELISA, and the assay has a much higher sensitivity than a usual ELISA, and this allows us to capture those originally neuronal proteins even in the blood as needed for trials. So the hypothesis or the research questions we were asking is, are neurofilament light chain and neurofilament heavy chain, are they elevated in SCA3? And if so, do they rise before the symptomatic stage in humans and before the symptomatic stage in mice? And if so, when before the symptomatic stage do they arise? And finally, what is the relation of blood NFL and PNFH to ATXN3 aggregation? Those are the questions we try to ask in humans and, if possible, in mice. And in this talk, given the time limits, I mainly focus on NFL. The work is published now. There you can also read more about PNFH, but I will mainly focus, due to reasons of time, on NFL. For the humans, we had two separate cohorts. Cohort 1 was SME plus Tübingen samples. We call it SME plus cohort. 160 patients and NFL, a neurofilament heavy chain, were assessed by the commercially available Quanterix SCA kit. As an independent, so to say, validation cohort, just to confirm whether our findings are really replicable, we used the EUROSCAR-RISCA cohort, 89 subjects, and a slightly different assay, again a SCA assay, but it was a homebrew assay capturing NFL and PNFH. So it's two different cohorts with a slightly different assay, arguing if we couldn't replicate our findings in the other cohort with a slightly different assay, it's really a robust finding. That was about the humans. How about the mice? In the mice, we used a very recent mouse model from Janette. It's a 304Q-in ATXN3 mouse model. We investigated heterozygous and homozygous mice in the pre-symptomatic and in the symptomatic stage. And here we used again the homebrew NFL and PNFH multiplex on the SCA. I start with the findings in the humans, and I start in the first cohort, this SME plus cohort, and I start with the ataxic SCA3 patients. You see them in red, you see in blue the controls. What you first of all, on the first glance, you see that NFL, like PNFH, is related to age. So you see an increase just due to age, your brain degenerates. But what you could also appreciate is that in the ataxic SCA3 subjects, they're always constantly above the age-matched controls. So NFL is increased in the ataxic SCA3 subjects compared to controls. How about pre-ataxic subjects? You see them in green. Also, the pre-ataxic subjects are always above their age-matched controls in blue, except one, but that was probably a pre-ataxic SCA3 subject way before clinical onset. So those were an example of the single subject data from the subject number 1. Now we come to the group data of cohort number 1. You see that in pre-ataxic subjects, NFL in green is increased compared to the controls, and also the ataxic subjects are increased compared to controls. In this cohort 1, we did not find a difference between ataxic versus pre-ataxic subjects. How about the second independent cohort with a slightly different SCA assay? We could replicate the finding that in pre-ataxic subjects, NFL is increased compared to controls. And here you also see that in ataxic subjects, it's even higher than in pre-ataxic subjects, arguing that there might still be an increase in the ataxic stage. Looking at this more closely, we were correlating NFL with the SARA score, and we saw at least a moderate correlation between the SARA score and NFL levels. So this argues for the fact that you might want to use NFL as a disease severity fluid biomarker. Could you also use it to stratify subjects according to disease progression? So we did a median split between the high versus low progressors as defined by the annual change in the SARA score. And yes, at least with a certain effect range, NFL does distinguish between the high versus the low progressors in SCA3. Summing this up, this might point to the fact that NFL might serve as a treatment response marker, namely if you could renormalize the NFL levels. We'll come back to this at the end. And it might serve as a subject stratification marker in upcoming SCA3 treatment trials. For example, if you want to filter out high progressors versus low progressors and stratify your clinical cohorts according to progression rate and want to have a molecular readout for this. So what is driving this increase of NFL, what we see in SCA3 subjects? There were two main factors. One is, of course, age, as I've shown you before. The second factor are the CAG repeats. And you see here a model which shows that it is age, but also the length of the CAG, which drives the NFL increase. So those are two of the main determinants of the individual NFL levels. So how about the pre-ataxic subjects? Looking at them more closely, what we were interested in is when related to CAG-based expected onset, when does NFL increase? And what we did in order to calculate this is we set normalized each individual pre-ataxic subject's NFL level, normalize it relative to the healthy controls you see here in blue. And then we calculated for the pre-ataxic subjects, when does the 95% confidence interval of the pre-ataxic subject no longer overlap with a set score of 2 from the healthy control? So when it's more than set score of 2 above that range. And what you see, first of all, is that you see in the pre-ataxic subject, you see a linear increase relative to its expected proximity to disease conversion. So it's kind of a proximity marker. And when you see is that the confidence interval does not overlap anymore with a set score of 2 to controls at about 7.5 years to expected onset. So you have a distinction 7.5 years before expected onset. So this points to the fact that you might want to take NFL even for stratifying your pre-ataxic SCA3 subjects to enroll them in trials, because you don't likely want to enroll any pre-ataxic subject 30 years before onset, but really want to hit those at the pre-ataxic phase, and NFL might help you with that. Talking in terms of trials, can we do sample size calculations for just by reducing NFL? So not clinical outcome measures, as Massimo has told you, but in terms of NFL. What we did in order to calculate sample size calculation is we calculated the short-term stability of neurofilaments. And you have heard Thomas Clockhatter asking Winfried, what is a short-term interval for movement sensors? We did the same now for fluid biomarkers for NFL. How stable is it over time? And you see it for NFL as well as for PNFH, for controls, pre-ataxic subjects and ataxic subjects. And you see that in this short-term interval where we want, so to say, the biomarker to be stable, it's really stable. And this stable means there is no noise. And if there's no noise, this means it gives you a much lower variability. And that is, of course, helpful for calculating a biomarker-based sample size calculation. And the numbers are then really low and much lower than what you have seen from clinical outcome measures like SARA or whatsoever. For example, if you want to have a 20% therapeutic effect, but being aware, therapeutic effect meaning not in a clinical scale, but meaning a 20% reduction of the NFL elevation, the NFL delta compared to controls. So 20% reduction of the NFL compared to controls, you need 7.5 subjects per treatment arm. So 15 subjects for intervention versus placebo arm for PNFH, a few more for NFL. But still, the numbers are incredibly low if you take it compared to a clinical outcome measure. And this argues for the fact that you might want to use in your future trials NFL as a measure to calculate sample sizes. And you might even use this, for example, to run interim or exploratory trial analysis, because you need really a small amount of subjects and can you do with very small cost in order to see, am I heading into the right direction for an interim analysis in your clinical treatment trial. So that was the work about the humans. How about the mice? In mice, we ask the same questions. First of all, we wanted to know, is NFL increased? And if so, how is it related to the symptomatic stage of the mice? So we took the 304Q knock-in mice, and it has two major phenotypes. One is the weight phenotype, which comes first at about month 10 of the age of the mouse. And then it has a motor phenotype, which comes rather late at about month 15. And you see the control, the wild type in blue, in the heterozygous mice in red, the homozygous mice in orange. And what you see is already before the motor phenotype arises, the NFL is increased in the heterozygous mice compared to the controls. The same is true to the homozygous mice. So before the motor onset. Interestingly, in the late stages, this levels off, and it's not because NFL reduces in the SCA3 mice, but because the wild type ramp up. And that's just because the mice get older and the brain degenerates, and then NFL gets released. So you lose the effect in the aging mice. So how about, can we still capture by NFL the mice still before the weight loss, before any behavioral readout at all? So we look at the pre-symptomatic, so the pre-weight loss mice. And what we found is even there, NFL is increased compared to wild type mice in both heterozygous and homozygous SCA3 mice. At age month 2, there is no difference as of yet. So this means NFL starts to ramp up at about a month 6 in this mouse model. How is this related to histology? And I think the mouse model is excellent to tell us how is this related to what actually happens in the cerebellum. And in this mouse model, the Purkinje cell loss, and we were able to show this in this paper, starts about month 6. So NFL is even at the verge of the starting Purkinje cell loss. And this tells you have a peripheral readout of what is happening cellularly in your brain, in your cerebellum. So this is, I think, a cool story if you want to use the mice as a preclinical, NFL in your mice as a preclinical trial. The final question then is, how was that related, the NFL increase, how is it related to ATXN3 increases? And here I just showed the ATXN3 soluble ATXN3, but the aggregated was pretty much the same in the cerebellum. So not in the blood, but in the tissue, and extracted from the cerebellum. And what you see here, first of all, is that ATXN3 already ramps up at month 2, at the stage where you do not yet see any differences in terms of NFL or PNFH in the mice. So ATXN3 is definitely there earlier. However, what you can also appreciate is that ATXN3 then does not go further up. So while NFL goes further up, ATXN3 does not so. And this leads me to the hypothesis that we can use ATXN3 rather as a target engagement marker, whereas NFL could rather be a disease progression and a disease severity marker, whereas ATXN3 is more a target engagement marker. So I would combine both at the end. I think that is the direction we have to head to. We're trying to do the same now in humans. We have the NFL, and Janet is measuring the ATXN3 in human blood, so then we can tell you whether the same is true for humans or not. So we were then able to map a biomarker cascade in SCA3 mice. That's only about mice. And what you see here in red and in green is the ATXN3, and you see it has a high effect size on the y-axis. On the x-axis, you see the age of the mice, and you see the ATXN3, whether it's soluble or aggregated ATXN3 from the cerebellum, it goes up, has a high effect size from very early on, but does not go higher anymore, but pretty much stays the same. NFL and PNFH is still in the normal range in the early stage, but then ramps up, has however a high effect size, and both ATXN3 and NFL and NFH come much earlier than the motor or the weight phenotype. So to sum up, what we are trying to show is, the suggestion is that blood neurofilaments in ataxias, and I've used SCA3 as an example, might serve as biomarkers for disease severity, even in the pre-ataxic stage. It might help to stratify subjects, for example, for identifying fast versus slow progressors. It might help to identify pre-ataxic subjects which are close to disease conversion, so it's a proximity marker. It might help to reduce sample sizes. And the suggestion would, at the end, I think, put it together with like ATXN3 or the other ATXN2 or whatever we are going to be able to measure, and to capture an external degeneration as a disease progression marker, combine it with a target engagement marker. And finally, I think NFL and PNFH are good markers in preclinical mice trials. Thank you very much.
Thank you very much, Matthis, and thanks for being on time. So there are 5 questions. Oh, there's now a new one. We have few minutes to answer them. Let's very quickly go through them. I think the first one was actually answered during your talk. Is this blood or CSF?
Oh, can you read them out? I have to, there's quite a lot of, sorry.
First question is if the NFL levels are measured in blood or CSF.
Oh, only blood, and that's a cool thing. I mean, what trials CSF is good, but that's the real advantage. In CSF, we have been able to measure NFL and PNFH, I don't know, since 10 to 15 years. The cool thing now, it's being able in blood, but for this you need to see more. However, there are other platforms coming up where you might also be able to do this, but for now, see more is the way to go.
Okay. So Dr. Jardem from Brazil asks, NFL was correlated to SARA change that occurred before the NFL measurement. Is that correct? This was earlier in the talk where you were presenting the first cohort results.
Yeah. So, oh, that is true. So we measured, so what we did in terms of the SARA, we had the natural history data, or the SARA quotient, we made an annualized change out of the SARA change. So we annualized that, and this has helped us to calculate fast versus slow progresses by the annualized change. And then we checked whether this was, the NFL helped us to divide those two groups.
So how did you define low versus high progression? This is a question from Dr.
Yeah, that was just by a median split. So that was, we annualized change and median split to groups.
Right. Then applicability question to other SCAs. Can NFL be useful biomarker on other SCAs such as SCA1?
So to my knowledge, but I think Opal is going to have a little talk on this later in the biomarker talk. So my knowledge, I'm not aware of any human study. However, in our Journal of Neurology paper, it's Wilkie et al. 2018, we have measured a mixed bunch of repeat scars, and we will find it across that. But those in-depth studies, I think this is the first in-depth study in humans. But my suggestion, my hypothesis is, and this is why I'm taking this as an example, I would bet a good German chocolate that you will find it in the other, at least the multisystemic ones. I'm not so sure about SCA6 because the cerebellum, if you don't have the long tracts, I would be a little bit more skeptical about SCA6, but for SCA1, 2, 3, 7, and 17, I would bet a German chocolate.
Okay, one final question, and the rest we will leave to the coffee hour. So please, the people who ask the questions, don't go away, stay for the coffee hour. From Dr. Ashizawa, have you tried to correlate NFL biomarkers to neuronal loss in the mouse or MRI data in humans?
So at this stage, we have done it qualitatively. Oh no, it was even semi-quantitatively. What we have done in this IMBO paper, we have related to the Purkinje cell loss. And we found a weak correlation, but it was at this stage is our histological techniques. Well, still, it's always hard to quantify the Purkinje cell loss and do it in a very exact quantitative fashion, and even in histology. So our measures were not perfect. It was more semi-quantitative measure, but there we could show that really the NFL is ramping up at the onset of Purkinje cell loss. So that's in terms of histology. In terms of MRI, that is the study we are running with Gülen right now. and we're not only with structural markers of MRI, but in particular, Gülen is an expert also in MRI spectroscopy. And I think it will be really cool to relate neurofilaments to structural as well as spectroscopy marker. And then we can do the same cascade as I've shown you for the mice in humans related to those imaging markers.
Thank you very much, Matthis. Great work. Thank you. So let's move on to our next speaker, Dr. Leonard Petrucelli. He's joining us from Mayo Clinic in Jacksonville, Florida. And his group has been at the forefront of protein aggregation issues in various neurodegenerative diseases, and he's going to talk to us about ATXN3 proteins in biofluids.
Okay, good morning and good afternoon to my European colleagues. I would like to thank Gulen and Matthis and Tomas for giving me really a unique opportunity to present our work on ATXN3 proteins in biofluids. And somewhat just as an introduction, most of the work that has emerged from my lab has primarily focused on frontal temporal dementia and ALS. But we felt that there might be opportunities for us to somewhat go into the SCA space because of the work that we were pursuing. So in particular, just as a background, because this work was really fundamental and has implications for the data that I will be presenting, was back in 2011 when the hexanucleotide repeat expansion consisting of GGGGCC was identified as a major cause for ALS and FTD. It presented really a unique opportunity because of the fact that this intronic sequence, although it was intronic, could actually produce in a very non-conventional form of protein translation these unique repeating amino acids consisting of glycine proline, glycine alanine, and glycine arginine in the sense direction. And as a result, it was what we showed, as well as the colleagues in Dr. Dieter Edbauer's group, that these dipeptide repeats, in fact, do exist in postmortem tissue of only those individuals that have the C9 mutation. Not only do they exist in the sense direction, but they could also occur in the anti-sense direction. And the unique dipeptide repeats would be the glycine, the proline alanine, the proline arginine. So this led us and prompted us to interrogate whether or not we could actually, with these antibodies that we generated, could any of them be detected in biofluids from patients? And so, here is just a summary of the work that we've done through an international collaboration with many ALS neurologists who provided their samples. You can clearly see using an immunoassay detecting specifically the glycine proline that we can readily detect it in CSF. And this population of patients is broken down to asymptomatic and symptomatic patients. You can clearly see that even though these patients are not yet presenting with disease, that they had readily detectable levels of poly-GP in the CSF. So just based on this, it was it allowed us to conclude, although we did the correlations, that there was not going to be much of a prognostic signal associated with poly-GP and the severity of the disease. And so I won't show you that data. We did show in iPS neurons in collaboration with Dr. Jeff Rothstein from Johns Hopkins University that not only you can detect it in the media and in the lysate, but in this time course experiment in which we are collecting media following treatment at the respective concentrations of ASO, that you can readily detect a significant decrease in the poly-GP, with 50% remaining around days 10 to 15. So it does take some time, that even though the RNA is in fact hitting, the ASO is in fact hitting the RNA, it is takes some time for the poly-GP levels to get degraded, presumably because there really is no specific degradation signal such as lysine residues that might facilitate its degradation. I've also just included this because it was a preclinical trial in which we dosed our C9 AAV animals with ASOs, and while we saw a significant reduction in the number of RNA foci, which is not shown, but in the three dipeptide repeats here, GA, GP, and GR, you can appreciate the significant reduction in immunochemistry as well as biochemical analysis. But more importantly is that we were able to show a significant reduction in the CSF. So this work really prompted and somewhat set the framework which is also being included in a recent clinical trial for C9 ASOs, that you might use this a marker for target engagement. And so that led us really to, are there opportunities in SCA3 for us to potentially exploit? And since there is a poly-Q stretch consisting of the CAG repeat within the exon, we felt that might be an opportunity for us to somewhat use a similar strategy. You have a long stretch of these poly-Qs. Would we be able to generate an immunoassay similar to what we did for poly-GP? And so I think that everyone can appreciate that, as therapies for SCA3 are really emerging, what would be some of the criteria that would be necessary to really facilitate the development of these therapies? So target identification and validation, development of bioactive agents, means to diagnose patients in early, if not pre-symptomatic stage of the diseases. And similar to our ALS and FTD work, could we have a marker that would be able to assess the efficacy of treatment and the target engagement in clinical trials, especially for those that are RNA-based? So our work started 3 years ago. This is our Mayo Clinic Biomarker discovery team, headed by Dr. Zbigniew Zolik, who's been a movement specialist for several years now, Dr. Mercedes Prudencio and Dr. Rana Hanna. And they were absolutely critical in terms of facilitating our ataxia biospecimens collection. We didn't focus it only on SCA3, but we've also have been in the process of collecting other SCAs as shown here. And part of the processing includes collecting CSF, blood, skin, and also urine. So the initial assay development, we decided to use the mesoscale discovery platform, in which we are capable of using an antibody to capture an ATXN3. And then for our detection, we used in a sulfatag conjugated manner an anti-poly-Q. So one of the first steps that my laboratory does to develop these assays is really just screening the available commercial antibodies, if in fact that they're available. And so this is just shown here, a Western blot from a patient fibroblast, and you can appreciate that at least this one antibody that detects the total ataxin, that in the SCA3 patient, it can detect quite nicely the expanded as well as the wild type allele. In the control healthy patient, it detects both species, but under these conditions, it's impossible to resolve the two alleles, since they run at the similar molecular weight. We've also generated in an ataxin-3 protein. This is just shown here, recombinant, then run on a gel in a concentration-dependent manner, as well as taking that material and running it on a Western blot to show that the antibody used in the fibroblast experiment can readily detect it. And this just shows the standard curve and the level of detection. So in the first cohort, we call it is defined by the Mayo cohort. During that period of 2 to 3 years, we were fortunate enough to recruit a total of 54 patients, as well as other ataxia patients. Our ongoing C9 ALS collection served as a disease control for these studies, as well as healthy controls in a few pre-symptomatic individuals. And so this is just a breakdown of the patient characteristics for the samples that were used in our study. So we were also able to collaborate with Dr. Paolo Giunti. We felt that this was absolutely necessary to validate and support our studies by having an independent cohort of samples. And so this is the patient characteristics associated with the samples that were provided to my lab. So shown here using this poly-Q assay, you can see that we were able to detect really a very specific signal in the patients that have the SCA3 mutation carriers compared to healthy individuals. We were also able to see, albeit lower levels, some level of the poly-Q in the patients who were pre-symptomatic with the mutation. We were unable to detect really a specific signal in either the other ataxias as well as the C9 mutation carriers. When we looked into plasma, we found actually similar results, although the sensitivity of the assay actually was better. And you can see here that the poly-Q levels were readily detected, similar to what we found in CSF, likewise with the pre-symptomatic individuals and our healthy and disease controls. And using the validation cohort, we found similar results. So overall, what does this mean? The fact that you can detect it in both CSF and plasma, and does it have any clinical significance? So taking the features of the disease, including SARA scores, disease duration, and age of onset. To our surprise, we did not find, following multivariable analysis and multiple corrections, there were really no associations with the poly-Q levels with any of the disease features, including age of onset, disease duration, and what have you. Similar results were also found in the plasma series for both the combined cohorts. In that, we also included some additional clinical criteria that is used by some of the physicians. So what about NFL? We heard from Matthis just moments ago. And so we took the opportunity to measure NFL using the same platform, the Simoa Quanterix platform. And as you can see here, we can readily discriminate the patients with amazing specificity and sensitivity, which is not shown here. Occurs, but I think you didn't see, although samples were few, for pre-symptomatic cases that had modest levels of NFL. Likewise, in the plasma, we found that there were significant levels of NFL that can discriminate between the healthy individuals. And once again, there were some levels, although comparable to the healthy individuals. So what does it mean clinically, similar to the poly-Q? Will there be any associations found with NFL levels, given the fact that you can readily detect it and it's quite high in SCA3 patients? So following multiple multivariable analysis and corrections in our cohort, we did not see, surprisingly, any changes of NFL with age of onset and disease duration, as well as any of the clinical criteria, including the SARA score and the gait mobility. So as the next step, we took advantage of making sure that our cohort was in fact representative of other groups and other movement disease specialists. And so what we did here was we just simply looked at the length of the ataxin repeat and see if it associates with age, since that was previously shown and is well documented. And in fact, we can see that in our cohort that there is a significant association with the length of the repeat with age of onset and a modest association with SARA score. All these didn't reach significance, but they were trending in the right direction. So overall, the cohort of patient samples that were used in this study were consistent with the previous results. So we feel confident that the poly-Q levels as well as the NFL levels, despite not showing significance, are in fact reproducible. So we next brought our attention to whether or not we could detect this in patient fibroblasts, and this is just shown here, the process that's involved, and we can readily detect the poly-Q protein in patient fibroblasts using our immunoassay, and that those patients that we collected that had both fibroblasts and CSF, that there was a correlation between the two. So in this experiment here, what we did was we took several healthy controls as well as SCA3 patients and then treated them with siRNA, where we could knock down the gene of interest. And what this shows here is that as predicted, if you knock down the ATXN3 gene, you can significantly cause a reduction in the poly-Q in every line, patient line that was detected. So we next drew our attention in the next couple of slides here is that a SNP was previously reported to be associated with ATXN3. And this is an interesting SNP because it's 103 nucleotides downstream of the repeat expansion, in which a tyrosine is converted actually to a premature stop. And as a result, that premature stop now encodes a much shorter protein product that presumably is believed to be involved in increased aggregation given the repeat is included. And so what we did was we wanted to assess whether or not what is the representation of this particular SNP in our SCA3 population. And so this is the breakdown here in which we were able to show quite specifically that this SNP with the TAA is actually represented in the majority of the longer allele, meaning the allele that has the expanded repeat. That was also consistent, albeit the number of cases that we have for the pre-symptomatic individuals is fewer. Trends, were in the same, direction. So overall, we feel that this might be an opportunity for, gene-based therapies that can target a selective manner that the majority of this particular SNP is in fact in about, 80% of the expanded allele. And that might have opportunities since we do not know what the intended consequence might be for knocking down both alleles, both the wild type and the mutant allele with the repeat expansion. So in summary, we developed an immunoassay that readily detects the polyQ protein both in bodily fluids, and in particular CSF and fibroblast. It can discriminate SCA3 patients from healthy controls and other forms of ataxia, since it's an ATXN3 specific assay. It hopefully will serve to assess the variability in patient responses to ATXN3 targeted therapies. And the SNP that we pursued in identifying that it is on the expanded allele may represent new opportunities for RNA-based therapies. So with that, I'd like to thank many of the individuals, at least quite remarkable timing given the fact that SCA Global is having their meeting this week. And so I think hopefully, we will hope that our work is, well received. And for additional details of the paper, it'll be out this Wednesday. So with that, I'll stop, and if there's time, entertain a few questions. If not, I'll be available for the coffee break session.
Thank you very much, Dr. Petrucelli. We have about 2 minutes to go over a few questions. I would like to start out with this one because that came up for Matthis in the previous talk too. Do you have data or hypotheses about the source of the ATXN3 in the plasma? Do you think it arises from the CNS or peripheral tissues?
So when we did a correlation between the patients that we had both plasma and CSF, we didn't find a correlation between them. So that suggests to us that the pool of polyQ that we're detecting in the periphery is most likely coming from non-CNS tissues, unlike NFL. The NFL, I think everyone would agree that it's coming from the brain, since it's brain specific and making its way into the periphery through, axonal and neuronal injury mechanisms.
Thank you. Of course, I misspoke. The question for Matthis was about NFL, not ATXN3, but we'll go over that during the coffee hour. So what do you believe drives the difference in ATXN3 levels between pre-symptomatic and manifest ataxic patients? This comes from Dr. Scholz.
So, I'm sorry, can you repeat the question again, Gulin?
What do you believe drives the difference between pre-manifest and manifest ataxic patients?
In terms of our ability to detect polyQ proteins between the pre-symptomatic and symptomatic individuals, I think it's very consistent, with our ALS work in the C9 space, is that, the repeat is present, and so it is, producing that protein independent of the disease process. Maybe it might accumulate, you know, as the disease progresses, but I was not surprised that we were capable of detecting the polyQ protein in the pre-symptomatic patients as well, based on our ALS and FTD work.
Okay, thank you. Let's keep the other questions for the coffee hour, and let's change gears to the MRI session, for MRI portion of the session. So our first speaker is Dr. Marcondes from Brazil. He's been doing great work in SCAs, particularly SCA3, and he's going to present about SCA3. Go ahead, Marcondes.
So good afternoon to everybody. Thank you, Gulin, for the introduction. So it's a pleasure to be here in this session. And I'm going to discuss a little bit about MRI findings in Machado-Joseph disease, and with a particular focus in the potential use of imaging markers as outcome measures to track the disease progression. So, so no disclosures. So the studies I'm going to show you have been funded by Brazilian agencies and also FARA. And the main, learning goals of this talk would be, I think, two. So the first one, I'd like to review briefly the neuroimaging signature of SCA3, and then I think the most relevant part for the clinical use of these measures. So we're going to discuss a little bit about the potential usefulness of neuroimaging as a biomarker for clinical trials or for prospective studies. So just to begin, I think we all know that Machado-Joseph disease is one of the main autosomal dominant ataxias. But in addition to ataxia per se, so there are many other manifestations such as dystonia, Parkinsonism, pyramidal signs. So all these clinical features point to a widespread involvement of the brain in this condition. And indeed, when we come to see pathological data, it becomes quite clear that the cerebellum is involved, as you can see here in this slide, but there are many other regions which are also, relevant targets of degeneration in this condition. So I'm talking now about the cortex, about the cerebral cortex. I'm talking about basal ganglia, so some of them are also involved, and also structures such as the brainstem and the spinal cord. So knowing this profile of this condition, so when I think about neuroimaging, I think the major goals are to characterize and try to quantify in vivo the extent of this neurodegeneration, and with two main objectives: to understand the disease pathophysiology, and as I mentioned to you previously, to try to find potential biomarkers. So in this discussion, I'm going to focus on MRI. So I think this is, we all know that this is a non-invasive and widely available, method. Even considering the high field scanners, I think it's now much more, available, the 3 Tesla scanners and even the 7 Tesla scanners. So it provides an excellent anatomical resolution that includes brain and spinal cord, which are, of course, two relevant regions for this condition. And I think one important point about MRI is that you're able to obtain different kinds of data. So I'm talking about volume, I'm talking about white matter integrity data like DDI, and also neurochemistry, spectroscopy results. So a quick overview of what we have in terms of SCA3. So when you look at the data coming from voxel-based morphometry studies, looking at gray matter atrophy, it becomes quite evident that the disease is characterized by cerebellar atrophy and also involvement of the basal ganglia, as you can see here in these two publications. So this is, of course, one of the main targets in this disease, and it has obvious correlations with the major clinical features. But damage extends to other regions. So this is a paper from our group published some years ago, where we could find that the cerebral cortex is also involved in Machado-Joseph disease. And as you can appreciate here in this figure, this cortical involvement mostly involves regions around the precentral sulcus. So basically precentral and postcentral cortex. These are also two regions involved in this condition. Other relevant portion of the central nervous system involved in Machado-Joseph disease is the spinal cord. So we're going to discuss this a little bit more during the talk, but I'd like to call your attention to these findings. So this is a publication from our group also, where we were able to perform volumetric analysis of the cervical spinal cord in these patients. And as you can appreciate here, the values were different in comparison to control. So there was significant atrophy. And also you can appreciate here that the spinal cord, it flattens in the anterior posterior regions. Moreover, when you look at the comparison, the correlation with the clinical features, you can appreciate here that the cord area has a significant, I'd say moderate correlation with the SCA scores. Now moving a little bit to the white matter. So we now have plenty of data showing that the white matter is also involved in this condition. And so in this study, you can appreciate that not only fractional anisotropy, but also radial diffusivity are found to be abnormal in these patients. And I'd like to draw your attention to the involvement of the cerebellum, of course, but also of the peduncles, as you can see here in these images. Well, looking at the cross-sectional data, we also have some interesting, I'd say very interesting data about the neurochemistry in these patients. So spectroscopy has been really useful in these patients. And in this study published some years ago, you can appreciate the results of the concentration of NAA against creatine with voxels placed in the cerebral hemisphere and also in the vermis. So as you can see here shown in the red arrows, that these values are significantly different in SCA3 patients compared to the controls. So showing that you can also see, I would say, functional abnormalities in these patients revealed by spectroscopy. So these are all, I mean, very, I would say, initial results, these cross-sectional findings. And it's interesting that when we try to sum up all the results and try to look at the potential correlations of imaging markers against clinical scales, you can see that, I mean, several of these parameters have shown relevant correlations with clinical scales. So cerebral volume, brainstem volume, the volume of some of the basal ganglia, namely the caudate nuclei, the cord area, as you saw previously, and also some DTI parameters. So these are all initial results that point to the potential usefulness of imaging as an interesting area to be investigated as a source of potential biomarkers. Well, but moving on, I'd like to discuss other points. So this is something I find interesting. And this is a paper published a few years ago, and we tried to compare patients with Machado-Joseph disease that had different phenotypes. So we look at a patient that had dystonia, as you can see here, and patients that had no dystonia on clinical examination. And it's interesting to realize that when you look at the structural maps in these two groups, they are, of course, they have some regions that were similar, but we could notice some interesting differences, namely the involvement of the caudate nuclei, which was restricted to the group with dystonia. So this is interesting because it shows us that there are regions which are, I would say, always found to be involved in this condition, but there are some other regions that could not be involved in every single patient. And this could be one of the reasons for the different phenotypes we often see in Machado-Joseph disease. Now, moving forward, I'd like to bring some data about at-risk individuals. So I'm going to talk a little bit now about individuals that have the CAG-expansion, but are not really truly symptomatic or not truly ataxic. So the very first paper that came out showing imaging results in this group of patients was published by the European group in 7 years ago. And in this paper published in the Lancet Neurology, the authors were able to show that the brainstem volume of SCA3 carriers, as you can see here, were slightly smaller than controls. So this was I would say, the very first evidence that structural damage could be found before the onset of ataxia in these patients. So more recently, other studies came out and actually gave further support to this initial assumption. So this is one publication from our group. It came out 2 years ago, and we were able to recruit more than 90 patients with Machado-Joseph disease. And we're able to stratify this cohort actually into 5 groups. So the first one, which is this S1 group, included 12 subjects that were actually pre-ataxic carriers. And then we stratified the remaining patients, those with ataxia already clinically evident, into 4 different groups. And these groups were defined according to the disease duration. So group D2, up to 5 years of disease duration, group D3, from 5 to 10, D4 from 10 to 15, and group D5 with disease duration longer than 15 years of age. So, and then we're trying to evaluate and compare the structure of signature in each of these distinct subgroups of the disease. And I think interesting results came out. So when you look at this initial figure, you appreciate that in patients, in the non-ataxic mutation carriers, we were able to find some regions with damage. So namely within the brainstem and also in the basal ganglia. And then when you look at the different groups stratified for disease duration, you can really appreciate that this is kind of spreads, and then it extends to the basal ganglia, it extends to the brainstem, and in the later stages also to the cerebral cortex. So this really suggests that the disease actually somehow begins in the lower portions of the brainstem, and then it spreads. It goes up to the remaining structures of the brain, involving in the late stages the cerebral cortex and the subcortical white matter. Looking at the cerebellum, so we had also some nice results. So as you can appreciate, in the first 2 groups, we could not see, any relevant change in terms of the cerebellum, but in the third group, we noticed the initial damage in the cerebellar lobule stand, which then spreads to the cerebellum overall in the last 2 groups defined according to the disease duration. When you look at the spinal cord, we also had some nice results. So this is in blue, you can see the controls, and in orange, you can see the patients stratified for the different subgroups. So it's interesting to realize that even pre-ataxic patients had smaller spinal cord areas, and these values tend to go down as the disease progressed. So even though this was a cross-sectional study, it presented some nice results in terms of the staging of the disease, considering patients across all the disease stages. Nice results in the same profile of patients came out using spectroscopy. This is a publication from the group of Dr. Goulinos. So they were able to evaluate 21 patients with Machado-Joseph disease and compared against 22 healthy controls at 70. And it became quite clear that looking at the cerebellar vermis, cerebellar white matter, and the pons, that these patients had actually reduction of the NAA concentrations. And this is quite evident here when you see the dot plot here. So again, showing that patients begin to present structural or functional abnormalities quite early in the disease course. So now we're going to move a little bit to show you some data, some longitudinal studies. So I think this is the very first longitudinal study, including patients with Machado-Joseph disease. It was published 7 years ago and actually scanned 19 patients using a 1.5 Tesla scanner and with an interval of almost 2 years. So the main findings here were actually the volumetric reduction of the putamen in the caudate nuclei found in these patients over 2 years. The second study I'd like to show you is a study from a French group. It was published 2 years ago in the Image Clinical, and in this study, authors were able to evaluate a cohort of 20 patients with Machado-Joseph disease, and the follow-up here was again 24 months, so close to 2 years. So in this study, authors actually looked at volumetry and also at the DTI analysis, but the DTI analysis were just cross-sectional. So I'm going to focus on the prospective data, and I'd like to show you the findings reported in this study about the normalized volume of the cerebellum and the normalized volume of the pons in patients with SCA3. So as you can see here, authors were able to find a progressive decline of the volume of the pons and the cerebellum in these patients. And they also made a step forward and tried to compute the effect sizes of these imaging parameters in comparison to clinical scores. So in brief, I think the main finding here is that brain volumetry data had a much higher effect sizes when compared to clinical scores, namely the SARA score and the CCFS score. So finally, the last study I'd like to show you is a study published recently. So this is a 5-year longitudinal study. So our group was able to evaluate 23 patients with Machado-Joseph disease in this long-term follow-up. And we also looked at diffusion tensor imaging analysis and also gray matter analysis. So in brief, what we found in this study was that we're able to find a progression of atrophy in these patients and also progression of white matter abnormalities in these individuals. And again, we were able to find that the effect sizes for some of these parameters were much higher than they were for the SARA scale. So when you look at the effect sizes for the cerebellar volumes, you can see here like 2.05, 2.07 for some of the lobules, and also for some diffusion tensor imaging markers, like you can see here for the inferior cerebellar peduncles, 1.2, 1.1. So this data point to the potential usefulness of imaging as an interesting biomarker for Machado-Joseph disease. And we look at this map here, which I brought from a publication, recent publication from Dr. Leonard. So where are we now in terms of using MRI as a potential biomarker for Machado-Joseph disease? So we certainly have some nice data considering the biomarker discovered, as I showed you previously. I would say we already have some validation in terms of reproducibility, in terms of sensitivity to different disease stages, but we definitely need additional clinical validation. And this is going to be only possible if we move forward and try to design, large cohorts, because this is indeed a rare disease. So this is something the field is moving, I would say, quite fast. So we now have at least 3 large international consortium that include SCA3 as one of the diseases of interest. So the ASME consortium, the Enigma Ataxia, and the REDISCA. So many countries and many centers involved in these studies. And the main focus, I think, are basically 2. So to identify and validate imaging markers, especially for patients in the early stages and even in the pre-taxic stages. And also something which I find quite challenging is to harmonize MRI and MRS protocols across multiple sites and across multiple scanners. So just to give you a flavor of what is going on, I'd like to bring you a very initial results from one of these consortium. You're gonna see more details in a presentation during this conference. So this is a cross-sectional result from the ASME consortium led by Jennifer Farber and Thomas Klocketa. So authors were able to collect data from an impressive cohort of patients. So more than 200 subjects and almost 50 pre-taxic carriers. and more than 60 controls. And in this initial evaluation, authors actually focused in brain and spinal cord volumetry. And overall, the main findings were that the spinal cord, the brainstem, and pallidum were all found to be atrophic already in pre-ataxic subjects. And more interestingly, they were able to find that such volumetric reduction begins 20 years before ataxia onset. So something really interesting and something which is really promising in terms of use of imaging as a biomarker. So just to summarize and to conclude, I'd like to give you some messages, some final messages. So first, the signature in terms of imaging for SCA3, I think the major targets are the cerebellum and the peduncles, brainstem, basal ganglia, and thalami, cerebral cortex, and the spinal cord. And at this time, I think my potential imaging candidates would be basal ganglia volume, namely the putamen and caudate volumes. I believe that cerebellum and pons volumes. And from our last study, this long-term follow-up, it seems that DTI would be something really interesting, especially focusing in the cerebral peduncles. I'd like to end by thanking my group, especially Thiago Junqueiro, which has been someone really, essential in our group in terms of the analysis, in terms of the protocols. I'd like to thank my collaborators in Brazil and abroad, and our sponsors, our funding agencies in Brazil. So thank you.
Thank you very much, Marcondes. We are out of time. I will allow one question. Oh, Thomas is trying to say something. Go ahead, Thomas.
Comment. I think it's now quite clear that the effect sizes of volume changes are in the same range or even higher than the changes of SARA or other clinical outcome markers. But this depends on the stage of the disease. And you mentioned this cross-section study. What we've seen there is that in the pre-symptomatic stage, the curve may be very flat. And then at the beginning of the disease, there's a steep decline. And then again, with progression of the disease, for example, the pons volume does not lose further volume. So these comparisons might depend on the stage in which you do the comparison. And in particular, in very late stages, the clinical outcomes may be more sensitive to change than the volumes.
Yeah, I think this is absolutely correct. And I'd like to just to give an example of that, which is the spinal cord. So when we look at that very first paper from the Annals of Neurology, we look at the spinal cord and we could find that it was perhaps one of the first regions to be abnormal in these patients. But in this last paper, the 5-year follow-up study, we could not find actually significant change. And this was indeed a very long-standing cohort. So I really believe that the choice of the region and the choice of the parameter is going to depend on the stage of the disease. And this is something really important in terms of the design of the study overall.
Yeah, this is exactly what we find in the cross-sectional study where you also contributed, in which Jennifer Faber will present tomorrow. So the spinal cord is small already 20 years before the onset of ataxia, and there's not much further decrease. Seems to be even a developmental problem rather than atrophy development with the spinal cord in SCA3. So this is my interpretation.
Right, thank you, Thomas. Thank you, Marcondes.
Thank you.
In the interest of time, we'll save the other questions that were all technical anyway to the coffee hour. So last but not least, it's a pleasure to introduce my dear colleague, Dr. Pierre-Gilles Henry, who has been leading MRI and MRS efforts in Friedreich's ataxia together with Dr. Christoph Langlet. So he's going to tell us about that experience. Thank you, Pierre-Gilles.
Friedreich's ataxia is the most common ARCA, and multiple MR studies have shown substantial cross-sectional difference in the brain and in the spinal cord of Friedreich's patients versus controls. And a small number of single-site longitudinal studies have also shown MR longitudinal changes in Friedreich ataxia patients over time, suggesting potential value as MR biomarkers. And I'm mostly going to show you results from the UMN Fares study, where we followed an early stage Friedreich's ataxia cohort over time with 28 subjects. And as you can see, the disease duration was only 5.6 years, time from diagnosis only 2.3 years. So it was really an early stage cohort. And we scanned 88 patients at baseline, 21 at 12 months, 19 at 24 months, and 14 at 36 months. And we also had 20 control subjects that were age and gender matched, but these controls were scanned only at baseline. We had a multi-model MR protocol with a Siemens 3D, Prisma, and in the spinal cord, we acquired T1 and T2 for morphometry, MRS for spectroscopy and DTI, and in the brain, also T1 and T2 DTI and QSM focus on the dentate nucleus. And mostly in this talk, I'm going to show you results in the spinal cord, which is technically more challenging, but with a recent progress in MR scanner technology, it is feasible to acquire even spectroscopy and DTI data in the spinal cord. For spinal cord morphometry, we use SpineSec, which was developed in Brazil in Marcondes' group to manually segment the spinal cord. And we found very large atrophy in Friedreich's compared to controls, and also longitudinal changes at 12 months and 24 months. For spine DTI, we use automatic segmentation using spinal cord toolbox and then FSL to determine the diffusion parameters. Again, we found a large reduction in FA compared to controls in FRDA patients, and longitudinal decrease in FA. And at the same time, we found an increase in mean diffusivity in Friedreich's patients versus controls, and a longitudinal increase. And we saw a similar increase in AD and RD. And for spectroscopy, we found also large differences in patients versus control, and the changes were actually large enough that they could be seen on individual spectra from patients. When we take the ratio NA over minocytol, we found that there was almost a 50% decrease in the ratio NA over minocytol in patients versus control, and there was almost no overlap in the values, with one exception. And there was also a longitudinal decrease in the ratio over time. So if we look at all the data together, you see that the changes could be as large as almost 10%, 5 to 10%. The diffusion parameters, diffusivity, were around 9% per year. These are the annual change from baseline. The ratio NA over minocytol was minus 7% per year, and urea at minus 5% per year. And if we look at the individual metabolites, NA and minocytol, we found that it's mostly NA that decreases over time, and minocytol tends to be stable. But because the SD on the individual parameters is much larger, so it was not significant, whereas the ratio, taking the ratio really removes a lot of instrumental sources of variability, and it was a lot more reproducible and statistically significant. And if we look at if we extend the longitudinal progression that we see at V1, V2, V3 to our baseline, that we see that we expect a large difference between Friedrichson control, even at disease onset, and probably a lot before onset. That could reflect a neurodegeneration that occurs many, many years before onset, but that could also reflect the fact that the spinal cord may not develop completely as normal in Friedreich's ataxia patients. So lessons learned, the large cross-sectional differences just a few years after onset suggest that Friedreich's ataxia is likely both a developmental and neurodegenerative disease. For example, in the case of spectroscopy, it's likely that the minocytol is not normal due to development, abnormal development, whereas NA reflects more the neurodegeneration. And that means that if and when asymptomatic patients are routinely identified in the future, MRI could help track hypoplastic and neurodegenerative changes even before the onset of symptoms, and that would help decide when to start treatment. So I can see a large role for MRI in trying to answer those questions of when to start treatment during development. Second lesson, the multimodal protocol provides multiple and complementary information on disease progression, with morphometry providing information on atrophy, and diffusion imaging on more microstructural changes, and spectroscopy on biochemical changes. And third, the longitudinal changes that we observe in the spinal cord. We also observe changes in the brain that I did not show here, but the changes in the spinal cord in Friedreich's ataxia tend to be larger, up to 5–10% per year, than in the brain, which can be 1–3% per year maximum. I remember this is an early stage cohort, so we don't know yet if this applies to later stage. But we think that it's important to include spinal cord MR measures as biomarkers in Friedreich's ataxia, not just the brain. And that shows that the MR protocol must be tailored to each ataxia depending on the main sites of degeneration. And fourth, as I said, the most sensitive MR markers of disease progression likely depend on disease stage. For example, it's possible that the spinal cord markers could be more sensitive in early stage and brain markers more sensitive in later stage. So there is a strong need to characterize MR biomarkers on larger cohorts, allowing stratification. And therefore we need prospective multi-site studies, because this is the only way to reach a large number of patients. And so towards that goal, we are participating in a new multi-site prospective longitudinal neuroimaging study that will have 307 subjects, 200 patients, and 107 control scans at baseline 12 months, 24 months. There will be 6 academic acquisition sites, a harmonized protocol that will include both brain and spinal cord, as well as clinical, cognitive, mood, speech, and blood biomarkers. And enrollment is going to begin soon, by the end of 2020. The main sponsor is FAR, with academic sites being Melbourne, Minneapolis, Aren, Campinas, Philadelphia, CHOP, and Gainesville with the University of Florida. We have industry partners, Pfizer, Takeda, Voyager, Novartis, and Exico, and collaborators for the blood and speech assessments, UPenn and Redden Lab. And finally, I'd like to thank my colleagues and thank these organizations for funding, especially FARA, FARA has funded most of the longitudinal study in Minnesota. Thank you very much.
Thank you very much, Pierre-Gilles. So I think we are going to catch up with the time we have lost. There is one question from the audience, from Dr. Helen Puccio. She's asking about the average, range of the age. The average age was 19, I believe she says, but what was the range?
The range, yeah, the range was age 10 to 30, mid-30s, 35 approximately. And we didn't go below age 10 because of the issues with motion, with MRI.
Mm-hmm.
I have also one question. I'm not allowed to put anything in the Q&A, so I always have to start to dive in. Pierre-Gilles, this is fascinating, your spinal cord data. I'm just a little bit more cautious about doing the extrapolations about the developmental stage. In particular, like you have done this, like this linear extrapolation of those 4 FA patients and have done a linear extrapolation. And I would be really careful because we have seen in ALS, FTD, but also SCA3 that this never works. The disease course is completely different, the pre-symptomatic in the late stages, as we've shown for NFL. So I wouldn't do a linear track there.
Yeah, we can be sure until we actually do studies at early age. But as you know, it's not possible at the moment to identify patients with asymptomatic patients in Friedreich's ataxia.
But maybe actually just to follow up, we had this in the last virtual coffee room. We were just thinking with Massimo Pandolfo and Laura Khadimos on the virtual coffee room. We were just thinking whether we might want to use ARCA Global and do a survey and see, would we be able to get pre-symptomatic FA patients, meaning mainly the siblings of the affected ones, and will we be available as a consortium to get a feasible number of that? And I think that will be one of the projects we might want to push forward with ARCA Global. Let's see whether we can get some subjects.
Yeah, so very good point. Thank you. Yeah.
Follow up on the age range question, I would like to get this one in. Did you see more changes in the earlier onset group, or did you look at any correlations in the markers with the size of the GAA?
Our cohort is a bit too small to do stratification by age, especially as we go to follow up. We have only 20 subjects. Well, I don't think I could still look at the data. I haven't looked separating the early onset and later onset. Yeah, I will let you know on future talk.
Thanks to all speakers for a great session.